All right, thank you all for joining us and welcome back. And thank you, our directors, Tracy and Becky, for setting the stage for today's main event, the Professional Internship Program. I would like to kick off this morning by introducing you all to one of Jordyn Anderson's mentors, Dr. Carla McDermott. Dr. McDermott is a professor in the Department of Marine Science at the University of Hawaii at Hilo, our very own home. Carla is famous for her love of limu and her expertise in the field. Her passion for marine science and science education is contagious. She has mentored many TCBS graduate students over the years, both through thesis and internship routes. Please join me in welcoming Dr. McDermott to the Zoom microphone.
Thank you, Lisa. That was too long an introduction. Today is really to focus on the students. And before I start, we're going to turn you first talk to the ocean. So next year, I want to hear not just a land acknowledgement, but also an ocean acknowledgement. And Jordyn will do that. She did that through her research. I first met Jordyn in the fall of 2019 when she had just arrived from Manoa. And she said she was interested in limu. And I said, great, I need somebody to help me teach a workshop in a couple weeks on seaweed identification and hands-on with specimens and microscopes. And she said, great. So I took her. I didn't know her. I didn't know if she even knew what a species was. And she did great. She was a great teacher. She was fearless. And oh, by the way, it was fourth graders. Then she decided she wanted to work on a project to combine not just working for an agency, but she wanted to do some of her own experiments. And she has created a very interesting professional internship. She worked with Ocean Era, and I see that her mentor, Keely Martin, is also here supporting her. Thank you, Keely. This semester, I was lucky to have Jordyn as a teaching assistant, well, really a partner, a team teacher in a lecture class, which is unusual for a graduate student instead of just a lab class. But I thought that it would further her experience and her career. And my goodness, she is a quick study. She's organized, she's fun, she's caring. The students love her. She has a command for the information, and she taught me an awful lot. And I personally am going to be sad to see her go. The department is sad to lose her, but we are also so happy that she has completed her master's. And so, Jordyn, I know you're eager to share with us your knowledge of the ocean and of the seaweeds. Take it away.
Thank you so much for those kind words and that lovely introduction. I'm going to share my screen. I get a quick thumbs up that looks okay, Lisa. You're looking good? All right. So thank you again, Carla, for that introduction. I wouldn't have wanted anyone else to introduce me here today. Aloha to everybody else. Thank you all so much for being here. Before I get started, I do just want to say thank you to Carm and Kevehi for presenting our Oli this morning, and also the land acknowledgement, which we'll add the ocean in eventually. Thank you to Lisa and Becky and Tracy for setting the stage this morning and introducing our program. I'm really honored to be starting us off with the presentations this morning. As you've heard, my name is Jordyn Anderson, and I'm here today to tell you all about my internship experience here in the Tropical Conservation Biology and Environmental Science program. I want to congratulate my cohort members, the ones presenting with me here today, as well as the ones that will have the pleasure of hearing this upcoming fall. I couldn't be more proud of you all. Now, the focus of today's talk is titled Assessing the Reattachment Potential and Large-Scale Aquaculture Feasibility of Native Hawaiian Macroalgae. Some of the species that I worked with, both in my experiments and at Oceanera, which is my internship agency, are pictured here. On the left here, we have a fragment of Amansia glomerata, which shows some really interesting results in my reattachment experiments. And the two right-hand photos are of Halimenia here, and then myself holding some Halimenia, one of the species grown at Oceanera that I can just never seem to take my eyes off of. I think it's one of the most beautiful plants. I have a pressing of it hanging right above me as I present here today. This work would not have been possible without the support of my committee, Ms. Lisa Canale, the internship organizer, Dr. Carla McDermott, who oversaw my experimentation work, and of course, Keely Martin, my mentor at my internship agency. And I'll talk about that in just a few slides. So speaking of those next slides, here's what you can expect from today's presentation. To start, I'm going to first bring us all up to speed on native Hawaiian algae, or do the best that I can in the limited time that we have here today. Once we're familiar with the plants that I had the fortune of working with, I'm going to walk you through the experiments that I conducted to explore the reattachment potential of different species of native algae. To conclude, I'll introduce you to the organization and share with you my time at Oceanera, where I not only learned practical aquaculture skills, such as caring for species of red algae, like a Gardiella, which is pictured here, but also practiced what I like to call the art of writing standard operating procedures for a living, working aquaculture facility. And it wouldn't be my time as a graduate student without sharing with you quickly my time as a teaching assistant. So, to understand why research on native algae is so important, it's key that you understand why these species are at risk. Unfortunately, there are a suite of threats that are mounting against many species of algae within the Hawaiian Islands, and really around the world for that matter. There's no doubt that ocean conditions are changing. Temperature and acidity are on the rise at global scales, and at a more localized scale like we see here in Hawaii, conditions are changing even more drastically due to high inputs of nutrients from sources such as runoff and sewage. This photo here in the bottom right was taken off the coast of Maui, where inputs of nutrients are incredibly high due to sewage and cesspool drainage. This is also a problem that we see really prevalently here on Hawaii Island. You can see this cloudiness or murkiness in the water, and that is actually the seeping of these high nutrient waters through the bottom of the ocean and into this nearshore reef environment. Increased nutrient loads are not alone in their siege against native species, as pollution also proves to be a formidable foe. Camilo Beach is famous worldwide for its accumulation of physical pollutants, and we find that right here on our own island, pictured here in the bottom left. I'm sure Mike will tell you all about Camilo Beach in the fall. So while trash is not consumable to algae, it still poses a threat to the ecosystems that they are a vital part of. What I would call the most dangerous threat to native species here in Hawaii is invasive species. Just like Kavehi is going to tell us about later on this afternoon of the terrestrial threat of invasive species, there's also invasive species of algae that are here, and they unfortunately outcompete natives. In the top right photo is Kaneohe Bay over on Oahu, where species such as Capophycus and Yucuma have taken over entire reefs and are fittingly called the smothering seaweeds. Fortunately, those two species aren't commonly found here on Hawaii Island, but there are a suite of other invasives that pose a huge threat to the native assemblages of algae, which our reefs hold, and they help to keep in place a really delicate balance that the ecosystems have. If you know why algae species are threatened, it's also important that you know why it matters that we protect these species, why we conserve them. Broken down to the three simplest descriptions of why native Hawaiian algae are important. We note that 1, they're a food source, 2, they hold cultural significance, and 3, at their most basal level, they still are primary producers. You might not realize this, but algae account for producing half of the world's oxygen. So for every other breath that you take, you have an algae to thank for it. But let's pretend for a moment that providing us with the most necessary element on Earth, that being oxygen, let's pretend that wasn't enough for these species. Why else do we care? Well, here in Hawaii, not only are these native species an important food source for reef fishes, as well as our honu, the green sea turtle, which we all know and love, many of these species are also an important food source for the people of Hawaii today, and they have been especially important historically. In addition to being a food staple to the people, some of these species also hold significant places in the culture to represent forgiveness, such as limukala or a sargassum species. These plants have great healing abilities, not only of physical wounds, but also emotional ones. And they're given to a party who has been wronged, and then the two parties eat them together as a sign of reconciliation. So we could talk really all day about the reasons why, but I hope for just these few that I outlined here that you can see why native Hawaiian algae or limu, why they're so important. So with that quick background of not only why algae are important, but what threats they face today, let's jump into the focus of my internship projects. If you split my internship experience in half, here is the first half. And that was completing an experiment with Dr. McDermott of the Marine Science Department as my mentor. We brainstormed for what I felt like was hours on what would be an important question to ask for my master's internship. And we actually decided to build on the work of a previous tropical conservation, biology, and environmental science student, Miss Cecile Walsh, and further explore the reattachment potential of native Hawaiian algae. Ms. Walsh did this back in 2014 with a thesis that was titled Reattachment and Transplantation of Native Hawaiian Macroalgae, a potential tool for reef management and alien algae mitigation. In that, she tested 22 different species of algae to better understand their reattachment potential. So, building off of this work led me to my primary question, and that was, does fragment size impact the likelihood of reattachment in 5 different species of native Hawaiian algae? As you'll see in the coming slides, my project expanded to explore more than the originally decided 5, but for the purpose of this question and purpose of the results, we're going to focus on those 5. The leading null hypothesis would be that there is not an impact on the ability to reattach because of the fragment size for any of those species. So, the species that were included in my experiments can all be seen here. Something you should know about algae is that they can be divided into one of 3 phylum. Phylums being a major taxonomic grouping, so large that humans and cats actually belong to the same phylum, right? So very large groupings. So with algae, there's 3 major phyla. There's the rhodophyta, which are the reds, the chlorophyta, which are the greens, and the phaeophyta, which are the browns. And in this experiment, we made sure to include at least one representative from each of those 3 groupings. So given their names, you may have been able to deduce from these original 5 species, which are on the left. My red representative is pictured farthest to the left here. This is Pterocladiella capiculi. The top of my second column here is my second red representative, which is Amansia glomerata. In the bottom of the second column is my phaeophyte, or my brown representative, being Podina. My green algae were represented by 2 species. At the top of the third column, we have Caulerpa lentilifera, and the bottom of that same column here is Microdictyon, which is a really cool, really net-like green algae. Also photographed here are the 2 species that I worked with that were grown over at Oceanera. They were included in my study, but not for the entire duration. The species are Halimenia, which is at the bottom of this column here, and then Agardiella, which is all the way on the right. So I could spend my entire allotted time here walking you through how I did these experiments, but instead we're just going to go through the most basic steps. So most of the plants that were used in this experiment were collections from the wild. I did them myself. They are from either Richardson's Ocean Park or Onekahakaha. Both really popular snorkeling spots here in Hilo. If you're on island, you probably know where they are. I would just like to note that when I collected these plants, I collected them as sustainably as possible, in that I took only the plants that were needed for my experiment, and when I harvested them, the holdfast was left behind. If you're not familiar, the holdfast is basically the algae's equivalent of a root system that we see in a terrestrial plant, so it allows them to attach to the substrate and grow. So when you collect a plant and leave behind the holdfast, the plant has a chance to regrow from the same point. So that occurred for all of my wild collections. Species that were not collected from the wild came from one of 2 aquaculture facilities. As I mentioned previously, there was 2 species from Oceanaria that got added during my experiments, Halimenia and a Gardiella. The third being Caulerpa lentilifera, which was the final aquaculture plant that was provided to me after being provided to Dr. McDermott from the Pacific Aquaculture and Coastal Resources Center, or PACRAC, which is the aquaculture facility right here in Hilo. Once these plants were collected, they were given 24 hours to acclimate to the experimental conditions, which were set up in 5-gallon buckets with a single air stone and a covering of plastic wrap. The plastic wrap was not actually initially in the experimental protocol, but it was added to keep out bugs and lizards, but it only did part of its job. We did this experiment during prime termite time here in Hilo, and I came in one day with an entire layer of termites covering my buckets. So we went along and added those. I also mentioned lizards. I did have 2 lizards that got into these buckets and died. And while I don't necessarily have the data to support it, I do think they impacted my experiments. But regardless, after the 24 hours of acclimation in the buckets, the plants were divided into their appropriate size categories. They were attached via raffia, which is a natural fiber, 2 pieces of rubble that was coral. So the topmost photo here shows myself tying a piece of Caulerpa, that green algae, to the rubble by wrapping the raffia around twice and creating an X-shaped pattern over the plant. And that was standard to attaching all of the plants to their pieces of rubble. These were then set into the buckets. The air stone was put in the middle and it was turned on, and they were left outside the Wentworth building on the UH Hilo campus. The bottom right photo is the standard setup for these buckets with the 6 randomized pieces of algae attached to the substrate around the edge, with a single air stone in the middle, which was weighed down by a rock so that it didn't float to the surface. They were left there for a total of 3 weeks, and at every week the water was changed and the buckets were completely cleaned out. After the 3-week period, the raffia was removed by cutting it on the opposite side that the algae was attached to, and then carefully removing that raffia. And then after what I'm calling a gentle yet firm shake, one quick wrist movement, if the algae was still attached to the rubble, it was marked as reattached. If it did not remain, then it was marked as not attached. Some of the plants attached extremely well, others not at all. They didn't even dry. And then there were those that were kind of an odd case, just like Amansia that I mentioned in my first slide, which actually did the strangest thing and reattached to the raffia fiber itself rather than the rubble substrate. So let's take a closer look at some of those results. I'm going to orient you first to this figure. So on the y-axis, or the vertical axis, we have the number of samples. And for this figure, the horizontal axis, or the x-axis, shows the size category of the algae. And this is for all of the species that I used grouped together. In green, or in the mint color, we have the number of plants for each size category that did not reattach. And in blue, we have the number that did reattach. So what this figure displays is actually that the experiment showed us that size does not impact the likelihood of a species to reattach. The statistical analysis that I did were chi-squared tables using Minitab 2020 software. They supported this conclusion, but I did not include the numbers here in the figure. If the species were going to reattach, they were going to do it regardless of size. But as you can see with this second figure, where now the x-axis has changed to species, not all of the species actually reattached successfully. The colors represent the same thing. So green means it did not reattach, blue means it did, and our y-axis still remains sample size. And while you can see here, the sample sizes are a little uneven due to the difficulty of consistently finding some of these plants, which is just one of the realities that I learned doing these experiments. Seeing that not all of these species consistently reattached is actually pretty interesting to me, because all of these species were chosen because they all had successfully reattached in Ms. Walsh's previous work that I based these experiments off of. So while I recognize there are many things about our two experiments that were different, I think it just goes to show how much still needs to be uncovered about the reattachment process in algae. This may not have been what I was expecting. There's still a lot of value and implication in these results. So seeing that, you may be asking yourself, what does this mean? Jordyn is here telling me it's important, but why is it important that we understand how algae fragments reattach? Why they reattach to pieces of rock or pieces of string, and what are the next steps in this knowledge? One of those future steps could be ensuring that outplanting is as successful as possible when it's implemented. Reattachment of algae is important because without it, the plants that are outplanted on reefs could just as likely be swept away with the tide and out into the open ocean. Outplanting of native species is something that we see done time and time again here in terrestrial systems. On this island, ohia and koa are outplanted in forest reserves by the hundreds to help recuperate forests. Why can't we do the same thing for our undersea forests? During reef recovery projects, cultures of algae can either be outplanted or maybe transplanted onto reefs to help rebound population. Reefs in Hawaii just do not have the algal communities that they once did, for some of the reasons that I talked about earlier. And one of the ways we can help those populations is to physically put the plants back on the reefs ourselves. Particularly after cleanup events or removal of invasive species have been done, there's this gap that's left in the ecosystem. And because invasive algae or invasive species are usually the first ones on the scene, so to say, they're usually able to repopulate the area even if they were just removed from it. But we're able to remove the invasives and then outplant fragments or maybe even entire thalli, which is just a fancy word for the structure of the plant, of these native species. If we're able to outplant those on the reefs, then maybe we'll give them the head start that they need to be able to reclaim the reefs that they once flourished in. This has been done by some groups on Oahu with species that are particularly enticing to our honu, again, the green sea turtle, to make sure that they have something to snack on when they're in the area. If we understand the minimum size that is required for a plant to successfully reattach, or the conditions needed in order to attach in the shortest time frame, then we can ensure that we're creating the largest stock of plants that will be successful when outplanted on the reefs. What this experiment demonstrated is that there's not necessarily a critical size necessary when you're growing these plants for reattachment scenarios, but there may just as well be other factors that impact this likelihood. Size just doesn't seem to be one of them. Now, before I move on to my time at Oceanera, I just want to point out one thing. I designed this experiment based off the work of a previous TCBES student. And the results, I like to think, open the door for future students or even just other future scientists to really get to the bottom of what factors have an impact on reattachment potential. So now, let's switch gears and see how I spent the other half of my internship time. Oceanera is a Kona-based aquaculture facility that not only grows finfish, but also native Hawaiian algae. And with a grant through the Department of Energy, is exploring the potential of these species to be grown at a large scale in a novel offshore array. Formerly named Kampachi Farms, Oceanera created the state-of-the-art offshore pen off of the Kona coast as part of the Valela project, which is pictured here on the right. This was really the first of its kind. It was an unanchored net pen for the aquaculture of finfish, and back in 2012 was named one of the best inventions of the year. They've continued projects focused on offshore production since then, and as previously mentioned, are funded through the Department of Energy to explore the feasibility of an offshore array for growing native Hawaiian algae. The prototype for that design can be seen here on the left. But before algae can be put on such a large array like this, a lot of information about growth rates, reproductive events, even just durability in these open ocean conditions has to be explored. And this is where my work at Oceanera fits into the organization's larger picture. I spent my time here with Ms. Keely Martin, the LIMU technician on the project, who herself is a marine science, Hilo marine science grad. A significant portion of my time in Kona was spent learning the ins and outs of an aquaculture facility. A lot of time was spent cleaning and calibrating the tanks to make sure that the algae are getting the proper conditions needed in terms of cleanliness, water flow, light, and space. The two photos on the left of this slide here are what the before and after of a tank cleaning would look like. You can see in the furthest left photo is a Halimania tank. Started out looking less than ideal. There's a ring of other types of algae and diatoms growing just under what is the standing water line when the tank is full. But with a sponge and a little bit of elbow grease, I got the tank looking better in no time, as you can see here in the middle photo. In addition to keeping tanks clean and keeping these algae happy, I was also able to be a part of working through some trials of algae reproduction. Here in the right-hand photo, you can see me looking just happy as a clam, painting zygotes of sargassum onto a rock. And you did hear that correctly. I was painting them like with a paintbrush, one that you would just get from Home Depot. So after we've collected these zygotes, or the baby algae, we strain them out using really, really fine sieves till all the water is strained out and we have a really thick paste that is actually best picked up with this paintbrush, and then it can be spread across rocks. Then the rocks are left in a tank or multiple tanks, depending on how many there are, where they're protected from the sun with shade cloths to prevent scorching of these little tiny plants. And incrementally, they're given nutrient-rich deep sea water to facilitate their growth. In my time here at Oceanera, I got to see this entire process, from the attachment of these zygotes onto rocks with the paintbrush, all the way through until those plants grew up and actually grew their own reproductive structures, which was a great experience. The day-to-day of an aquaculture site also includes constructing things that you need with the materials available, as well as repairing those that are around you. Here are a few of the projects that I put together during my time at Oceanera. This bottom right-hand photo here is one of the drainage standpipes, a barrier for that I created for a tank of ulva. Ulva is a really leafy green algae. Think of wilted lettuce, and it's really easily broken apart. This barrier was made in order to keep the algae from coming into contact with the pipe that had a really strong suction from the drainage line. So that barrier was therefore a protection from the plants so that they would not be ripped up. In the top right, you can see that shade cloth that I mentioned in the last slide, which not only did I get to repair a few of, but make a new one during my time at Oceanera. This involves sewing fishing line through the holes of the mesh around this PVC frame to ensure that the plants don't get sunburned. The middle photo here is some air lines that are found in some of the experimental tanks at Oceanera. The black tubing along here is the porous air line, which without support would float to the top of the surface of the water. So I worked on making and attaching these PVC frames so that the air line sits along the bottom of the tank and appropriately aerates the plants. This last photo here on the left shows a stand that I made that would allow the filter bags to dry out completely after they're cleaned. The filter system became a focus of my time at Oceanera, as I'll explain in just a moment, and this stand was my very first introduction to it. This is actually one of the first things that I did on my first day on site at Oceanera. Little did I know how important the filters would become. This photo shows the stand while it's not in use, but once the filter bags are taken out of the system, they can be rinsed, bleached, and set upside down on the stand to ensure that they're able to dry out completely before being used again in the next cycle of cleaning. So the filter system on site at Oceanera became really important as it allowed me to update and improve upon the standard operating procedure, or SOP, for cleaning it. Not only did I learn how to clean the entire system myself, but I created a detailed document with photos to ensure that anyone who came on the site could clean the system should that be necessary. I did this first by writing up the procedures, then walking through and taking photos of each step and creating labels for the valves, creating labels for the pipes to ensure there was a continuity between the instructions and what is the physical on-site plumbing. Creating this document allowed me to contribute to the Oceanera organization for years to come as my time with them came to a close. So the photo here on the left shows the main pump and filter house for the entire algae site at Oceanera. For reference, here's a picture of as much of the algae site as I could get into a single photo. A fun side note is that this photo was taken from the top of the boat that Oceanera has that will hopefully soon be in the water taking these algae offshore. This photo highlights the array of tanks that are found on site. You can see these long blue tanks, which are the most plentiful on site, and they're often used for experiments because with so many, you're able to include appropriate replicates. The larger tanks in the foreground here are used to hold and grow the standing stocks of different species, so that there's always some biomass of them available. In the background, once we begin to get these round tanks, that is what begins and marks the fish site of ocean era. As I mentioned earlier, they're also a finfish aquaculture at this organization. The week this photo was taken, there's not many experiments taking place, which is why so many of the tanks are uncovered, because they don't have algae in them. When there's plants in them, they're covered by shade. Also, before I leave, just look at the location of this site. How could you not love this, right? If I remember correctly, during my entire time at Ocean Era, I only remember it raining one day. So you just can't get much better than that. But back to the focus, back to the filter system. So you can see that there's these two bays on the filters here, which use different sizes of filters to ensure the correct purity of water. During my time cleaning these, I really came firsthand to see how important they were. As one time when we cleaned them out, a snail had entered the filter system and released all of her eggs, which hatched into tiny baby snails. So we took them out and there was dozens, if not hundreds, of these baby snails, similar to those that can be seen here. At the top here is what would be the adult snail, similarly sized to the one we found in the filter bag that day. And then at the bottom is a similar size to one of those baby snails that, along with its brothers and sisters, all hundreds of them, coated the inside of these filter bags. If these had been pumped on site into the tanks of algae, they could have posed a major problem for the stocks of algae on site, particularly detrimental to the experiments conducting, which could be super dependent on set amounts of plants. So even the tiniest snail can eat a lot more algae than you think. The middle photo here shows the inside of the nursery container, where water is entering from the outside after being pumped and then is plumbed into various racks, bays, and tanks. This is especially one of the important places where I had to ensure continuity of labeling in the standard operating procedure I created to ensure that the valves get turned on and off in the correct order. You don't want to overfill any tanks or backlog any of the plumbing. It's a lot larger than it may look in this photo, as the width of the rack is more than doubled, and then it's also mirrored on the wall that is behind me. So it's really important that anyone that reads the SOP is able to understand what valves they should be looking for and where they might find them. The final photo here on the right shows part of the filtration system on the outside of that nursery container. This is just half of the filters that the water must go through before entering the nursery. And that includes running through a UV system, which you can see just a little bit of on the very edge of this photo. All of this filtration allows for the water to be as clean as possible, so that when rearing these really young plants and conducting experiments, there's less worry about contamination. And I say less because it's almost impossible to avoid completely, but we use all this to do our best and ensure that these plants have their best chance of survival, and really providing Ocean Era with that information needed to hopefully one day soon get offshore. So before I wrap it up here today, it would be impossible to describe my experience as a TCBES graduate student without acknowledging the opportunity that I was given as a graduate teaching assistant in the Marine Science Department. Pictured here, you can see me very diligently. My eyes look closed, but I promise they're not. Very diligently watching over some of my fall 2020 students as they took their final. This final, I was given some reference material, but was able to more or less design on my own. Over the 2 years here, I taught 8 sections of the Marine Science 171L, the Introduction to the Marine Biology Lab. And as Carla mentioned earlier, this semester, I got to co-instruct the 171 lecture with her. This experience has allowed me not only to grow as a student, but as a person and an educator. And I am eternally grateful for the experience that I got to teach in a real university setting. Adjusting my teaching to not only an online setting, but also what's pictured here, this hybrid in-person, ultra sanitized room with gloves. That's undoubtedly prepared me for the future that I really hope to find in education. I highly recommend that future students explore the possibility. It'll give you a lot to do. There's a never-ending pile of assignments to grade, but it'll be one of the most gratifying jobs you could have. When you get to take people snorkeling for your job and you find things like this amazing Spanish dancer that we did, it's really hard not to love what you do. So, to reiterate, I could not be more thankful for the opportunities provided to me throughout this internship experience. Lisa, thank you for your unwavering support of me as well as the rest of us internship students. Carla, thank you for taking me in as your grad student and allowing me to teach alongside you. Keely, thank you for being not only my mentor, but also a great friend. I couldn't have gotten to this point without the rest of my cohort, a few in particular. Thank you, Carm, for always making the drives to Kona super fun. To Andre for carrying my buckets of water when I asked, for listening to me talk about algae for hours on end, and just about everything else that you do. And to Dawn, I think you're here too, for not complaining when I asked you to clean my slimy buckets with me. So to wrap things up, I would like to thank all of you here on the Zoom, as well as those that are there on the live stream, for being here today and listening to my presentation. My contact information is in the bottom left corner. I'm always happy to talk about algae or just about anything else for that matter. And with that, I would be happy to take any questions. Mahalo for listening.
Thank you so much. That was fantastic. So if you have a question, feel free to type it into the chat. We have the time for 1 or 2 questions. Or if you're brave, you can feel free to unmute yourself and go ahead and ask it.
Lisa, do you want me to stop sharing or should I leave this here?
Oh, why don't you leave that up for right now?
Okay.
That'd be great. Awesome. Well, okay, I will, I just want to ask Tracy, she's monitoring the live stream. Tracy, do you have any comments there? Not yet. Okay, awesome. All right, so Jordyn, what I'd like to know is out of all your experiences that you had at the TCBS program, what do you think is going to be most informative in your journey that you're going to take forward in your career?
In my personal journey, given that I really do hope to find a future in education, I would say that my time being as a TA will really help build upon that. In the short term, I think having practical aquaculture skills for applying for different jobs and just looking around the possibilities of what I can find here on Hawaii Island. I am super grateful that I can say I know how to do some of those things. Basic plumbing, something I definitely did not know how to do beforehand. So yeah, short term and then long term, just having a start and getting to do some education on my own, say that I've taught X amount of classes, I think is going to be a real step up as I move forward.